Hello everyone, welcome back to my channel, Igcse Biology Help. In today's video, I'm going to be solving the May-June 2022 Paper 4 Variant 1 as requested by some of the students. So let's start solving. Question 1I. State the kingdom in which yeast is classified. As we all know that yeast is actually classified in the kingdom of fungus or fungi. You can write one of them. Fungus is actually the singular form, fungi is the plural. So you can write down either one of these. Double I, state the process that occurs in mitochondria to provide energy for yeast cells. Some students will write down respiration without mentioning that it's aerobic. No, you have to write down that it's aerobic respiration. This is how you get the mark. This is a 1 mark question, so please make sure to mention the word aerobic. B, yeast cells make the enzyme sucrase. Sucrase catalyzes the breakdown of sucrose to glucose. So this question is actually talking about the lock and key mechanism. So what does that mean? I'm going to explain it. This means that each enzyme fits into one specific substrate. So it's complementary to the substrate. This is the keyword that you have to write down to get the mark. And you also have to say that enzymes are specific to specific substrates. And what does that mean? It means, for example, if you have an enzyme that looks like this, you have to have a substrate that will look like this in this rectangular form. Of course, this is not accurate because I'm not really good at drawing, but you can see the concept. It has to fit like this, for example, just like that, into the enzyme. It has to be complementary. It has to be perfectly fitting into the enzyme. This is the meaning of the word complementary to the substrate. And you have to say that when it's complementary, you're going to have the substrate fit or bind into the enzyme or the active site or the sucrase enzyme. The students made an extract of sucrase from yeast cells. They investigated the activity of sucrase extract at different pH values. They determined the rate of the reaction at each pH. They then calculated the rate of each reaction as a percentage of the fastest reaction to give the percentage activity of sucrase. The results of this investigation are shown in Figure 1.2. Describe and explain the effect of pH on the activity of sucrase shown in Figure 1.2. Now, in this question, so many students will just write down the description of the graph over here, or they will write down explanation as of why the activity of the sucrase decreased after increasing the pH. No, you have to say both of the description and explanation. Over here, if you just describe, you're going to get maximum 4 marks. And you're just not going to get the full mark. You have to mention both the description and explanation. For this question, there is a couple of points that you have to highlight in your answer to make sure that you get the high mark. Now, you have to mention the peaks or the minimum values or the maximum values. What do I mean? I mean, for example, here you're going to mention in your answer that, for example, at pH 6, we have the percentage activity is at its highest or at its peak. At 100%, this is the maximum value. What about the minimum value? It is actually at pH 12 over here, as we can see, at the percentage activity of 10%. So this is what I mean by the maximum and minimum. Second thing that you have to mention is the trend, overall trend of the graph. What does that mean? It means that, for example, that if the graph is increasing or decreasing, or for example it is overall like increasing and then it decreased like we can see in this graph. So this is what I mean by trend. Third point is numbers. That means that you have to mention any, for example, at the pH of 9, you can mention that its activity was at 60%. So this is what I mean. You have to get a random like value on the graph and just mention it. You can say, for example, at pH 9, the activity was 60%. This is how you mention it in your answers. So these three points you also have to remember them in your answers. So first of all, you have to mention that from pH 2 until pH 6 over here in this range, the enzyme is actually working and it's active, and the rate of percentage of the sucrase is increasing, or the percentage of activity of sucrase is increasing. It is increasing because the pH is actually increasing. So at pH 6 it is at its highest, it is at its 100% activity. This is because there is the highest rate of enzyme and substrate complexes forming. This is the optimum pH. So this is why at pH 6 it is the highest. So I'm both doing the explanation and descriptions. This is how you solve this question. You just say this point, for example, is the highest because of whatever. So this is how you do this question. Let me complete. So now from pH 6 until pH 12 it is decreasing because there is the enzyme is getting denatured. That means that the active site can no longer bind with the enzyme. That means that it is, for example, if the active site was this shape, this was before it got denatured by increasing the pH. For example, the substrate would fit in like this, for example, but after denaturing, it kind of just like melted. So, for example, it became like something like that, for example. But this is not the same shape, of course. So this same substrate will no longer be able to fit into the enzyme. So the rate of activity, of course, will decrease. So this is why from pH 7, until 12, it is decreasing. So you can mention any of the points that I have mentioned now, and this is a very long question. It is 6 marks, so make sure to focus in it and solve it well. Question number 2, mammals have double circulation. Figure 2.1 is a diagram section through the heart of a mammal. State the name of the chamber of the heart with the thickest wall. Now obviously it is very obvious in this picture that the left ventricle is the thickest wall out of the other walls. Over here, this is very thick compared to, let's say, I'll do another color. This is the right atrium. As we can see, the right atrium is very thin compared to the left ventricle, which is in orange. So you have to write down it is the left ventricle. D is the vena cava. The vena cava is the vein that brings in deoxygenated blood from all over to the body into the heart. So we have to write down the description of its structure. As we all know that any vein has valves and it has a wide lumen and thin wall. You can write these down and you will get the 3 marks. This is just a question where you will mention the vein structure that we all know. So you can mention that down. And you can also say the wall is lined by single layer of cells. Also, if you want, you can explain, for example, why does it have valves? This is to prevent the backflow of blood. Wide lumen to prevent like the repulsion of the blood, so it doesn't like just go in with a high pressure, it goes in with a lower pressure. So if you want to explain, you can explain, but it is enough to just write down the structure, describe it. Identify the structure labeled X in figure 2.1 and state its role in the heart. Over here, X is actually the semilunar valve. Over here, we can see it. I'll just highlight it. This part. This is the semilunar valve. This is actually to prevent the backflow of blood, just like the valves. And this is to ensure that blood flows in one direction. This is 1 point. So if you say prevent backflow of blood and ensures that blood flows in one direction, this is going to give you 1 mark. They considered it as 1 point in the mark scheme. So this is question III. B, figure 2.1 is a diagram that shows the double circulation of a mammal. The arrows indicate the movement of oxygen and carbon dioxide in and out of the blood. Shade the blood vessel in figure 2.2 that transports blood with the highest oxygen concentration. So the highest oxygen concentration is actually from the pulmonary vein that is coming back from the lungs. Over here, it is this one. It is directly coming back from the lungs, so this is obviously the highest concentration of oxygen in there. So this is the answer. Describe the evidence shown in figure 2.2 that the mammal has a double circulatory system. Over here you can mention that the heart is like divided into two parts or two sides, and you can also mention that the blood flows twice in one circuit. A circuit means, for example, if it comes back from the lungs into the heart and another circuit from the heart into the other parts of the body. Explain the advantages of a double circulation. Over here, the double circulation will actually provide you with a higher pressure blood. That means that it will have more efficient blood supply to the rest of the body. We can also say that oxygenated and deoxygenated blood will be separate or kept separate. Also, provide a higher supply of oxygen to the rest of the body cells, and it will also provide any nutrient that you want. For example, you can say it can provide more iron, it can provide more vitamins, whatever you want. You can say that it will maintain a high rate of respiration. This is because the blood will come in with high rates of oxygen. This oxygen will be used in respiration, so when there is a lot of blood pumping into the body, there will be a higher rate of respiration. You can also say that it will allow more time for gas exchange, and there is a lot of points there's 11 points in the mark scheme. You can choose any of them. Question 3. The pancreas is an organ that has roles in the digestive and hormonal systems of the humans. State the names of organs J, K, and L. As we all know that the J is actually the liver, K is actually the gallbladder, L is actually the duodenum, or you can say the small intestine. You'll get the mark anyways. Complete table 3.1 by stating the hormones and enzymes that are secreted by the pancreas. As we all know that the hormones secreted by the pancreas is insulin and glucagon. Insulin is for increasing the blood sugar supplies, like increasing the sugar in the blood. Glucagon does the opposite, it decreases the sugar in the blood. Enzymes secreted by the pancreas is amylase, trypsin, and lipase. Explain how CFTR proteins move chloride ions across the membrane of cells shown in figure 3.2. The question says that the CFT proteins in the cell lining the pancreatic duct move chloride ions. So any ions will move by active transport over here. As we all know, there is carrier proteins in the active transport. So by mentioning the word active transport, you're going to get a mark. You're going to say, of course, the active transport process, it is against the concentration gradient, or you can say it is from low concentration to high concentration, and you can say that it uses energy. The protein changes shape. You can say that this protein is the carrier protein. So you can say carrier proteins over here, they will change shape to move the ions. So this is the active transport process that we all know. The movement of chloride ions into the pancreatic duct causes water to move from the cells into the duct to help the flow of liquid in the duct. Explain how water moves from the cell shown in figure 3.2 into the pancreatic duct. So the question is actually talking about water. So any water is actually osmosis. So we're going to be mentioning the idea of osmosis. So we're going to say that the water will move by osmosis down the water potential gradient, not concentration gradient. Concentration gradient is not going to be marked down as correct when you are talking about osmosis. You have to say water potential gradient. And you can say that the movement of water is through partially permeable membrane. Our next question is about genetics. If CFTR proteins do not move chloride ions, the liquid in the pancreatic duct becomes very sticky and the duct can become blocked. Blocked pancreatic ducts are one effect of cystic fibrosis, which is an inherited disease. So I'm just going to skip this down if you want to read it. The question is asking the allele that causes cystic fibrosis is a recessive allele. Describe and explain the evidence shown in figure 3.3 that cystic fibrosis is caused by a recessive allele. Over here, we can see that if it's black, for example, if it's a circular black icon, then it's a female with cystic fibrosis. If it's a square, then it's a male. So over here, we can see that there is two people over here in this whole family tree that have cystic fibrosis. And these kids, they are siblings. They come from parents that they don't have cystic fibrosis symptoms. So we can say that over here, the parents of 5 and 7, the kids that have cystic fibrosis are coming from parents that actually do not have cystic fibrosis. So we all know that this is caused by recessive allele. Recessive allele, again, is the allele that is not expressed. Only the dominant allele is expressed. That means if it is expressed, they will show the cystic fibrosis. But if it's not, then they will appear or show no symptoms of this disease. So we're going to say the parents are actually heterozygous or carriers of the mutant allele. Person 7 is expecting a child with a man who is heterozygous for cystic fibrosis. Complete the genetic diagram to predict the probability of person 7 and the heterozygous man having a child with cystic fibrosis. Use symbol A for dominant and lowercase a for recessive allele. Now we're going to write down the genotypes. This is actually, they said that the father is actually heterozygous, so we're going to write down A capital and A lowercase, and then we're going to write down person 7. Person 7 is actually 2 lowercase a's. This is because they have a recessive allele, they have it expressed, so this is a recessive allele. We're going to write down the letters that I just wrote in gametes over here. So we write it down in gametes because, as we know, that there is the X and Y chromosome, they are separate, so we write it down in separate boxes over here. Now we're going to do the crossing. We're going to write down A capital and A lowercase and 2 lowercase a's. We're going to cross it this way. You're going to connect the first letter with the second with the first letter in the second genotype. This is going to give us A and 1 lowercase a. We're going to do the same thing for the second letter with the first letter. This is going to give us 2 double lowercase a's. We're going to do the same thing again, but with the other letter. This is going to give us another lowercase and capital A. Again, we're going to do it with the lowercase a and the other lowercase a. This is going to give us 2 lowercase a's. Now we got the genotypes of the offspring. We can get, you're going to copy down the ones that we got, the results over here. You're just going to copy it down. So the phenotypes means the physical feature. We're going to say that they have with or without the cystic fibrosis. This is the phenotype. Probability, you can write it down in many formats. You can write it down as 50%, you can write it down as 1 in 2, or you can write it down as a decimal like that. It's up to you. Explain how protein in cattle manure is converted to the type of ions that plants can absorb. We can say over here that decomposers or bacteria or fungi will break down or decompose the protein into amino acids. The amino acids will be deanimated to produce ammonia or ammonium ions. So you can also write down the chemical form like that or like this. Also, we are going to say that the ammonium ions will be converted into nitrate ions by nitrification or nitrifying bacteria, and the ions will be used by the plants. It's just the advantage to farmers of having snap beans plants that have a large number of root nodules. Over here, they said that the root nodules contain nitrogen fixing bacteria. This, of course, will have an advantage to the farmers. This will actually make the farmers have more profit. This is because the plant itself has like the nitrogen fixing bacteria, so the farmers do not have to spend extra money, or they do not have to spend time to add the nitrogen fertilizer, and also they will use less fertilizer because the plant itself has the nitrogen fixing bacteria substance. It will produce more profit for the farmers, and also this will make the plant, of course, grow more efficiently and faster and better. Calculate the percentage increase in the average number of root nodules per plant when snap bean plants were grown with a large quantity of cattle manure, field 3, compared with no cattle manure, field 4. Give your answer to two significant figures. This is very important because they will actually deduct marks from you if you don't actually write it in the two significant figures as they requested. Let's have a look at the graph that is shown here. So over here in field 1, they said that it was treated with a small quantity of cattle manure, and field 2 was treated with medium quantity. Field 3 was treated with a large quantity. Field 4 was actually the controlled variable, which is not treated with any cattle manure. They wanted to compare field 3 to field 4. Field 3 had a large quantity, field 4 has not had any cattle manure in it. So they want to compare the percentage increase from the field 3 to field 4. Over here, for field 3, of course, will have the highest average number of root nodules. So we're going to get the values of field 3 and field 4 and calculate the percentage increase. Over here at 3, we're going to draw a line over here to help us identify the value. So this graph, we're going to divide it. You're going to divide 20 by 5 squares over here to get the number that is in each square. So when you divide it, it's going to give you 4. So actually this number is going to be, this is going to be 44, 48, and this is going to be 152, this is going to be 156. So the field 3 is actually 156. Field 4 is actually 96. So we're going to be calculating 496. So over here, we're going to calculate it by calculating the difference between the two numbers. You're going to get a calculator. 156 minus 96 going to give you 60. You're going to put the number before the change down in the fraction, the denominator. This is actually 96. This is because it was grown with no cattle manure. So we're going to put 96, and you're going to multiply it by 100 to get the percentage. Calculate this by your calculator, and you're going to get 62.5%. Of course, they told you that they want two significant figures, so you're going to write it down as 63%, and this is your final answer. When large quantities of manure are put on fields, it can lead to eutrophication of streams and rivers. This can lead to death of fish. Describe how eutrophication of stream rivers can lead to the death of fish. You're going to actually describe the whole eutrophication process, which is actually starting off from excess fertilizer being thrown into the rivers by leaching, for example. So when the excess fertilizer is thrown into the rivers, there is going to be algal bloom. This is going to cause excess plants going to grow on the surface of the water. This is going to cause the water to look or appear greenish in color. So these plants on the surface will actually block down the light reaching the plants down the surface of the water. The plants down the surface of the water will actually die, and when they die, the decomposers will go down and actually perform aerobic respiration to decompose the dead plants. This is, of course, going to take oxygen from the water. And when it takes oxygen from the water, the fish will die. So this is going to lead to the death of fish in the water stream. So this is actually the whole process. I just explained it right now, so you can read the mark scheme again if you want to study it. Over here they're asking the genus name of a species of a tree. You're going, of course, if you want to get the genus name, you're going to write down the first. Over here, this is the species name and the genus name. This is the species name and this is the genus name. The genus name is always the first word, and this is the second word. This is the species name. So we're going to write down this word, the first word. Just like that. And B, explain the undesirable effects of deforestation on the habitats that are on mountains, such as Mount Muljan. Deforestation means if the trees are being removed from a forest or any place. So when you are removing the trees, this is actually going to cause loss of habitat and therefore lead into loss of biodiversity. Loss of biodiversity can actually lead into extinction of some species. Actually, loss of habitat occurs because, for example, when you cut down a tree, some birds live on the tree, or some like unknown species of insects or anything will live on the tree that we don't see. For example, these plants or for example, animals will die. Also, there can be plants around the tree. This will all be like dead and removed. So this is going to cause loss of habitat. Also, when you remove the trees, this is going to cause the soil to lose its structure. It's not going to be as rigid as before. It's also going to cause flooding and landslides. Flooding will be caused because trees actually help to decrease the chances of flooding when there is rain in the forest. It will cause soil erosion because of the landslides and the floods. And there is many points in the mark scheme you can mention. And also you can say that it will cause visual pollution. Visual pollution will be caused because when you remove trees, the place will not look as good as before. As we all know that natural places look very nice and beautiful and relaxing. So when you remove the trees, it will not look like that anymore. Explain the benefits to other organisms on Mount Mulajani of the conserving the Mulajani cedar tree in its. As we all know that trees can actually help small animals to camouflage themselves or hide themselves from predators. So this is actually one benefit to animals for the trees if we keep them. And also it can provide some nutrients or substances that we can extract to produce medicines for humans, or we can use it in manufacturing of anything. We can also use it for decomposing. For example, the leaf litter will be decomposed to produce nutrients for the soil. And of course, the tree itself will be a food source for both animals and plants. D, suggest why it is important to collect seeds from many individual trees of each species rather than just one tree. Of course, as we all know that if we collect from only one tree, then the genetic diversity will be very limited. And let's just say that this tree has mutation or recessive allele, so it will be a problem to the future generations. So if we collect from more than one tree, then the future trees may be adapted to the changes in the environment because they have a high genetic diversity. They may also be resistant to diseases and pests, and they might not be able to carry the recessive allele or have mutations, and they may not be diseased if we collect from more than one tree. Question number 6. Figure 6.1 is a flow diagram showing the events that occur to form a human fetus. Complete table 6.1 by using the information in the flow diagram to identify the cell, the organ, and the processes shown in figure 6.1. Now, over here, they're asking about organ P. Organ P, let's have a look at the diagram. Organ P is actually converted into a diploid cell. So as we all know that diploid cells are produced from the testis or from the ovary. So over here, ovary is already written down. So of course, it's going to be the testis. Cell S. So let's have a look at cell S. Cell S is right over here. Diploid cell S. So actually, it is going to be the zygote. This is because the haploid sperm cell and the haploid egg cell have been fertilized together to produce the diploid cell S. So we are going to write down that it is the zygote. Process Q produces haploid sperm and eggs. Process Q, as we all know, it is the meiosis, not the mitosis. Mitosis produces the diploid. It's going to be meiosis. We're going to write it down over here. Process R produces diploid cell S. So let's have a look at process R. Process R is actually fertilization, as I just said from the previous question. It is actually fertilization because haploid sperm cell and haploid egg cell are going to fertilize to produce the cell S that I have mentioned, that it is actually zygote. So we're going to write down the word fertilization. Process T occurs so that cell S can grow into an embryo. So process T is actually going to be mitosis. Mitosis, not meiosis, mitosis. This is because, as we all know, that the zygote will actually perform mitosis to produce diploid cells to actually start to develop its cells and produce the organs. So it is mitosis. Process U occurs so that the embryo can gain oxygen and nutrients from their mother's blood. Let's have a look at process U. This is actually implantation. Why is it implantation? This is because, as we all know, that the embryo will actually implant itself to the fetus in order to get nutrients and oxygen and food from the mother's blood into the fetus blood. So this is all going to occur if implantation has occurred already. So this is called implantation. B. State why it is important that the sperm and egg cells are haploid and not diploid. As we all know, if it's diploid, then it's going to produce 4 cells in the zygote, not 2 cells. And of course, all babies have 2 cells. So we're going to write down that it's to maintain the same number of chromosomes, and so the diploid number of chromosomes that does not double at fertilization. So this is the 2 answers that you can write down. So this is done. State the function of the jelly coat that surrounds egg cells. Of course, the egg cell has the jelly coat. This is the egg cell, for example, and there's the jelly coat around it over here. So this jelly coat actually prevents more than one sperm to fertilize the egg or enter the egg. So if there's one sperm that has entered over here, then the jelly coat will actually harden and it will stop any more sperm cells from entering, so that there is only one sperm cell that fertilizes the egg cell. So this is the description that you're going to write down in this question, double I. C, and this is the last question in the exam. This is complete the sentences worth 7 marks. So each word over here is worth 1 mark. Now let's read down. The placenta provides a large surface area for the something of oxygen. As we all know that oxygen occurs by diffusion, so we're going to write down diffusion, or you can write down exchange of oxygen if you would like. And carbon dioxide between the maternal and fetal blood. Dissolved nutrients also pass across the placenta. Examples of dissolved nutrients are something, something, and something. So over here you can write down any 3 nutrients of your choice, and you can write down glucose, or you can write down amino acids, you can write down fatty acids, you can write down vitamins, anything of your choice. I'm going to write down fatty acids, for example, and amino acids. So antibodies pass from the maternal blood, giving natural Of course, passive immunity, as we know that the maternal blood actually give down passive immunity, not active immunity, and passive immunity to the baby for some infections that the mother has or has been vaccinated against. Each different type of vaccine contains one or more antigens taken from the pathogen or microorganism. You can write down, of course, most of the students wrote down pathogen. So it is taken from the pathogen or microorganism that causes the disease. And this was the end of this exam. What do I think of this exam? I think that it is very average exam. It is not that hard or not that easy if you have been solving many past papers before you entered that exam. Make sure to subscribe to my channel and like my videos in order to see more of this type of content, and good luck with your exams.